Okay, so hello everyone. I'm going to be doing the final FRQs now, but I'm gonna be doing them on my computer because for some reason my AP Classroom on my tablet will not open. So I'll do them on my computer. Okay. أصلا it's like kinda better because it's a bigger screen, but I just can't write. So you're gonna have to like imagine my amazing drawings on the screen. Okay. So describe the importance of the inner membrane separating different regions of the mitochondria and chloroplast. Guys, you guys just have to say what's it called? It just permits it allows different reactions to occur in different parts of the cell, or like you can say that it stops interfering reactions from happening at the same time. Just you know, just but I say stick to the what's it called? It allows different reactions to occur in different regions of the cell. That will that will make it like that will like give you the safest option. Based on the diagram, describe how the structure of these organelles controls movement across the cell membrane. Okay, so how the structures of these diagrams, how the structures of these organelles actually, what's it called? Control the movement across the membranes. You can just have to pick one of these. You can either say the membrane selective permeability, which I think is the best. Okay. You can say compartmentalization, you can say surface area to volume ratio, and you can say active transport. Why would I recommend membrane selective permeability? Because it controls what comes in and out of the what's it called, out of the membranes. So that's just that's just the thing that makes the most sense. Okay, so I just go with membrane selective permeability. Okay. Obviously we know how to find the volume and the surface area. We've done it 1,000 times. I also did this question before. Okay, so you just find the volume, you find the surface area, you do it in the ratio. You put volume over surface area. This is obviously the volume of a sphere and the volume of a no, just the volume of a sphere. They're just spherical. Okay, so you should be fine with that. Okay. Explain how the folding of the mitochondrial inner membrane and many individual chloroplast thylakoids are beneficial to a cell that's carrying out chemical reactions. I said this in the last video, so we say that the folding increases surface area, and that allows the membrane to hold more enzymes, and that increases the efficiency because what's it called? Because you can have more reactions happening if you have a bigger surface area and whatever. Okay, I said this so many times. Okay, this is one of the easier ones. This one I did it in the periodic video. Okay, so I'm not gonna do it again. I did it in the periodic video. You guys can just have a look at the marking scheme. Okay, so this is the question, B and C. This is A. Okay, this is A. This is B. Okay, this is part C. This is part CII. Okay, I did this in the periodic video last time. If you guys want the explanation, just go to that video again. Okay. Okay, let's start with number 55. I also did this one in the periodic video, so I'm not going to be repeating it. I want to make this video as short as possible, so I'm not going to be repeating that question. Okay. I did this in the first periodic video, but I did it wrong because I didn't know how to get the marking scheme, so I put it into AI like an idiot, and the AI gave me the wrong answer. So I'll be doing it again. Okay. Identify the process used to form the covalent peptide bonds that join amino acids into a polypeptide. So obviously we know that the process is synthesis, dehydration, condensation, whatever. Pick one of the names: condensation or dehydration, synthesis, whatever you guys want to put. That is the process that takes singular amino acids and group them into a big polypeptide. Okay. Part B. The change in amino acid sequence illustrated in figure 1 caused a change in the shape of receptor X. Okay, so receptor X changed shape. Okay, based on the R groups of the original and substitute amino acid, explain why receptor X changed shape. Ya Allah. What's it called? Here, if you can see, first we had in part A we had like polar side groups. Look, here are the side chains where my mouse is. These are the side chains. Okay, as you can see here that the side chains were polar. Okay, and then when the mutation occurred, what's it called? The polar side chains How do we know that it's polar? Because they're charged. Look, they're charged. The polar side chains lost their polarity. Okay, we substituted them for things that are non-polar. Okay, that's why. What's it called? That's why it changed shape. Using the template below that represents the 4 levels of protein structure, place an X on each level that is expected to be altered as a result of the amino acid substitutions in figure 1. Okay, this one you only have to put what's it called? You only have to put an X on the primary structure and the tertiary structure. Primary and tertiary structure. Why? Because if you read the question, scientists are studying the structure X, single polypeptide that's found in the membrane. Where is it? Okay. Contains no alpha helices or beta pleated sheets. Okay, no alpha helices or beta pleated sheets. That means that the secondary structure will not be altered. Okay, so using the template below, 4 levels of protein structure, place an X on each level that is expected to be altered by the amino acid substitution. Obviously it will only be the primary structure and the tertiary structure, because the quaternary structure is the many polypeptides together. Okay, and you don't have many polypeptides together, you just have what's it called, you just have one polypeptide. Okay, a single polypeptide protein. Look, they told you a single polypeptide protein. So to have a quaternary structure, you need more than one polypeptide. So that is why the quaternary structure also will not be affected. Okay, so it's just the primary and the tertiary. Explain how the amino acid substitution shown in figure 1 is most likely to affect the function of receptor X. Obviously, receptor X won't be able to undergo its function anymore, because we changed its shape. The shape changed. That means it can't bind to its what's it called, to its target molecule. The target molecule can't bind to it. Okay, and what's it called? It won't be able to undergo its function. So the response indicates that because function is determined by shape, the function of receptor X is likely to change, or the structural change might alter the binding of the molecule to receptor X. I told you that. Or the structural change might alter the ability of receptor X to cause a cellular response. I basically inferred that. Okay, so choose one of those. I say go with the structural change might alter the binding of the molecule to receptor X, but you can choose any one of those three. Okay, so number 57. Based on figure 1, describe 2 pieces of evidence a researcher could use to support the claim that apicoplasts evolved from free living prokaryotic organisms. Okay, so they're just talking about the endosymbiotic theory. Here you can see that this is a big cell, and inside this big cell there are multiple organelles. Okay, this organelle is called the apicoplast, and the apicoplast has DNA. Since the apicoplast has DNA, okay, it shouldn't have DNA because it's not a nucleus. Okay, so since the apicoplast has its own DNA, that is a sign that it evolved from something else. Also, it has a double membrane. Why do you have a double membrane? If you have a double membrane, that means you were engulfed by something. You remember those drawings that I always do, the engulfing? Okay, you get 2 membranes. So the double membrane and the DNA are an indicator that it was that it evolved from something, from a prokaryotic organism. Okay, then doxycycline is a drug used to treat malaria and some bacterial infections. Doxycycline binds to the prokaryotic 30S ribosomal subunit and inhibits its function. Okay, explain how doxycycline can be effective treatment for a P. falciparum infection in humans. So let me go down to the marking scheme. Okay, so let me just read this to you so it will be more clear. So the eukaryotic protozoan parasite P. falciparum is the causative agent of malaria. So that's a P. falciparum. P. falciparum cells contain an organelle called the apicoplast. Apicoplasts synthesize precursors of biomolecules that are required for growth and reproduction of the parasite. Okay, so since this doxycycline is a drug that is used to treat malaria, okay, this doxycycline will bind to the 30S ribosomal subunit and inhibit its function. So since we said that the apicoplast has its own DNA, if it has its own DNA, أكيد it will have its own ribosomes, because if it has its own DNA and it has a double membrane, معناته it evolved from a prokaryotic organism, and we know that prokaryotic organisms have what? They have ribosomes. So the apicoplast actually possess this, actually possess this 30S ribosomes, which means that the doxycycline, when it is used to treat the malaria, it will go to the 30S it will go to the 30S ribosomes, bind to them, and inhibit their function. When the function is inhibited, do you think that it will still be able to synthesize proteins? No, it won't be able to synthesize proteins anymore, and therefore it won't be able to grow, it won't be able to whatever, it won't be able to grow. Okay, meaning that شو اسمه, meaning that basically you killed the malaria cell in a way, like you stopped it from growing. Okay, so number 58, discuss the 3 properties of water. Guys, you know how much I hate the topic of water in biology, so I'm gonna let you guys pick whatever 3 you want of these. Okay, so you pick a property and you put its discussion. Okay, I've explained all of these like 20,000 times, so you should be fine on this. Okay, explain each of the following in terms of properties of water. You are not limited to 3 properties discussed in part A. Okay, so the role of water as a medium for metabolic processes of cells. It's what's it called? It's the wait, what the hell? Oh, okay. So I'd say go with the solvent, okay? Because that's what I always used to say. So the metabolic processes, the chemical processes, I always used to tell you guys solvent. It helps in the dissociation or ionization of materials, because you know how we took in chemistry, when you mix like a NaCl with water, you get Na plus and Cl minus and whatever. It helps with the dissociation and ionization of materials. That actually allows those materials to undergo chemical reactions. Okay, so I'd say choose solvent for the role of water as a medium for the metabolic processes. Then you say the ability of water to moderate temperature within living organisms, the high specific heat, obviously. It maintains the stable internal temperature. Then the movement of water from the roots to the leaves of the plants. This is where you can say what's it called? This is where I tell you guys to go with cohesion and adhesion, and the name of this fancy process that cohesion and adhesion produces, that cohesion and adhesion are responsible for, is the capillary action. The capillary action is like what's it called? It's like the movement of water inside the whatever, inside the xylem and whatever. Just say capillary action of water due to adhesion and cohesion. You know adhesion and cohesion. The only thing that you need to know is that adhesion and cohesion are responsible for this action. It's called the capillary action. You say in the written tomorrow, if this comes, you say that the capillary action of water due to adhesion and cohesion, because that's what I always used to tell you guys. Adhesion, cohesion for the root uptaking water and whatever. And yeah, that's it. Okay, so next question, number 59. In the provided model of a cell, draw arrows to describe the pathway for production of a normal CFTR protein from gene expression to final cellular location. In the provided model, draw arrows to describe the pathway. Oh, okay. So basically they're asking you about protein synthesis. We know, because we've said this a million times, we know that the nucleus has mRNA. This goes to the ribosomes on the endoplasmic reticulum to actually undergo protein synthesis. It undergoes protein synthesis. The proteins get transported along the endoplasmic reticulum, then leave the endoplasmic reticulum directly to the Golgi apparatus. The Golgi apparatus modifies the protein, packages the protein, folds the protein, adds any carbohydrates if it needs to the protein. It sends it off to the plasma membrane to be released to other cells and the rest of the body. Okay, so you can either draw these 3 small arrows, okay, from the nucleus to endoplasmic reticulum, from the endoplasmic reticulum to the Golgi body, from the Golgi body to the plasma membrane, or you draw one line right from the nucleus. But be careful from this line. It has to pass through the endoplasmic reticulum, then through the Golgi body, and then to the plasma membrane. Okay, be very careful. I recommend drawing the 3 small arrows. Well, it's way simpler, okay? Because this is very high risk, because somebody might draw an arrow من هون لهونيك هيك, passes nicely to the mitochondria. Okay, don't do that. Okay, if you know how to draw both types, I'd really recommend drawing both, because for some reason here they drew both. Okay, draw both tomorrow just in case. But if you can only remember one, don't risk this. Okay, because if you put it through here, it's wrong. If you put it through here, it's wrong. It has to go through this. It has to go from this, through this to that. Okay? Identify the most likely cellular location of the ribosomes that synthesize the CFTR protein. Okay? Obviously, we know that, what's it called? شو اسمه the ribosomes are either in the cytoplasm or they're in the endoplasmic reticulum. Okay, here they're in the endoplasmic reticulum, they're in the rough endoplasmic reticulum. Okay. Wait, but why? Oh my god. Wait, give me a second. Okay, guys, after consulting my private AP biology tutor, which is also known as AI, okay, I have come to the conclusion that, and I actually remembered it, okay, don't worry, this is like legit, I'm not talking from my head, okay. So we know that here, we know that what's it called, the CFTR protein is a gated ion channel. Basically the CFTR protein is in the plasma membrane, because the gated ion channels, where are they found? They're embedded in the plasma membrane. Okay, since it's embedded in the plasma membrane, okay, it has to be on the RER. Why? Because the ribosomes that are in the cytoplasm actually are only for things that are inside the cell. As long as you don't go outside the cell, the protein can be synthesized inside the cytoplasm. However, if you want to synthesize a protein for it to exit the cell or be embedded in the cell's plasma membrane, you have to go from nucleus to RER to the Golgi apparatus right to the plasma membrane. Okay, so that's why the CFTR protein is synthesized in the ribosome in the RER. Okay, so it's RER. Part C, identify the most likely cellular location of a mutant CFTR protein that has an amino acid substitution in the ATP binding site. Again, obviously it will be in the cellular plasma membrane. Why? Because the CFTR protein is a gated ion channel. Gated ion channels are in the plasma membrane. Okay, they are embedded in the plasma membrane. Even if it's mutated, even if it's whatever, it will still be in the plasma membrane. Okay. Yeah, I love this question. Okay, so the binding of an extracellular ligand to a G protein, so we're talking about cell signaling receptor, is the plasma membrane of a cell triggers intracellular signaling. Okay, after ligand binding, the GTP replaces the GDP, just like I said in my chapter 4 summary. Okay, you have to get GTP for it to be activated. So then now we have our GTP. Okay, that is bound to the GS alpha. Okay, so it's a type of subunit of the G protein. Okay, this causes the GS alpha to activate other cellular proteins. Okay, including adenylyl cyclase that converts ATP to cyclic AMP. Okay, so that's our secondary messenger. It produced the ATP to the cyclic AMP. The cyclic AMP activates the protein kinases. Okay, basically more like enzyme, basically like it's amplifying the signal right now. Okay, in cells that line the small intestine, a cyclic AMP activated protein kinase causes further signaling, amplifying the signal, that ultimately results in the secretion of chloride ions. Okay, so the secretion of chloride ions, okay, which is really bad. Okay, you'll know why in a bit. Under normal conditions, GS alpha hydrolyzes GTP to GDP. Okay, thus inactivating adenylyl cyclase and stopping the signal. That's the termination of the signal. Okay, I explained all of this in the chapter 4 summary, by the way, so if you don't understand anything from what I just said, you have to go watch the chapter 4 summary. Okay, individuals infected with the bacterium Vibrio cholerae, yeah, totally nailed that, experience severe loss of water from the body dehydration, so that is why it's bad. Okay, this is due to the effect of the bacteria cholera toxin that enters intestinal cells. Scientists studied the effect of cholera toxin on 4 samples of isolated intestinal cell membranes containing the G protein related signal transduction components shown in figure 1. GTP was added to samples 2 and 4 only. So we have 2 samples with energy, 2 samples without energy. Okay. Cholera toxin was added to samples 3 and 4 only. So we have the actual cholera thing in samples 3 and 4. Okay. The scientists that measured the amount of cyclic AMP produced, so we're trying to measure the amount of what's it called, of the cyclic AMP produced by the adenylyl cyclase in each sample. Okay, describe one characteristic of a membrane that requires a channel present for chloride ions to passively cross the plasma. Okay, this is part A. Describe one characteristic of a membrane that requires a channel be present for chloride ions to passively cross the membrane. What did we say? What is the one characteristic that we always talk about? We always talk about that the center of the phospholipid bilayer is non-polar. So it will not allow the polar ions to cross it, which means that it needs a channel for it to passively move through the phospholipid bilayer. Otherwise, it can't, because the center of the bilayer is actually non-polar. Explain why the movement of chloride ions out of the intestinal cell leads to water loss. Well, if I lose chloride ions from the inside of the cell to the outside of the cell, that means inside the cell my water potential is rising, and outside the cell the water potential is decreasing. And by the rules of osmosis, water moves from a region of higher concentration to lower concentration. So when I lose my chloride ions and the outside of the cell has less concentration of water, that means the water will have to move from the higher concentration that's inside the cell outside of it to the outside environment, which is the less concentrated environment, which is why we actually lose water from the cell to the outside. Okay? Identify an independent variable in the experiment. So the independent variable is something that we're changing. Obviously, it's either the GTP or cholera toxin. So we're adding it, we're removing it. على كيفنا, we're changing it. So the GTP or the cholera toxin will be our independent variable. It doesn't depend on anything. Okay? Identify a negative control. Guys, whenever you see negative control, look at something, whatever is disabled, whatever is معاق, يعني هون اللي قدامكم, yeah, choose it. So anything with a minus you can choose. You can choose sample 1, sample 2, sample 3. Okay? Please don't tell me I'm yapping. هيك. No, يا أخي, there's no way. Yeah, sample 1, sample 2, sample 3. Okay? What's it called? You can either say you can classify them instead of just saying sample 1, sample 2, or sample 3. You can say the samples without GTP, that's sample 1 and sample 3, or the samples without cholera, that's sample 1 and sample 2. Okay? And yeah, that's it. Or you can say the one that lacks both GTP and cholera, which is sample 1. I'm pretty sure that if you just say sample 1, you'll get the mark. But just in case, because they have it in the marking scheme here, okay, make sure that you write the one that's lacking GTP, both I and II, the one that's lacking cholera, both I and II. Sorry, the one that's lacking GTP is I and triple I. Okay, and the one that's lacking both GTP and cholera is I. Okay, so you can choose any one of those and go. Here's the marking scheme. Just choose one of these guys. Okay. And justify why the scientist included sample 3 as a control. شو اسمه? So basically here we have GTP and we don't have GTP and we have cholera. Okay, طيب. Control to compare cyclic AMP production of that of the sample having cholera toxin and GTP sample 4. Wait guys, give me a second. Okay guys, so I just realized that I'm stupid. They're not asking why sample 3 is the control. They're asking why the scientists use sample 3 as one of the controls. Basically, they used the sample 3 as one of the controls to compare it. So look here, sample 3, what does it have in common with any of these samples? Okay, so it doesn't have GTP, just like sample 1, okay? But sample 1 doesn't have cholera toxin, okay? In شو اسمه? In triple I, okay? شو اسمه? There is cholera toxin, and in sample 4 there's what's it called? There is also cholera toxin. But what's the difference between them? One has GTP, one doesn't have GTP, which is triple I. One does have GTP, which is IV. Okay, so they used sample 3 as a control, okay, to compare the cyclic AMP production, okay, what's it called, with a sample that has cholera toxin and GTP, and with a sample that has cholera toxin without GTP, okay. So to compare cyclic AMP production with that of the sample having cholera toxin and GTP sample, comparing sample III and IV enables the scientist to evaluate whether the activity of cholera toxin requires GTP. Okay, basically, okay, they said it in a similar way to me, but they included III and IV. So basically, by comparing the sample III and IV, you can then determine if the cyclic AMP production is due to the presence of GTP. Okay. What's it called? يا الله ليش خبصت. Comparing sample triple I and IV enables the scientist to evaluate whether the activity of cholera-toxin required. Okay. So basically, but when you compare sample 3 and you compare sample 4, okay, you can then figure out if the cholera toxin's activity is dependent on GTP or not. Okay, that's all they're saying. That's all they're saying here in this sentence. I say go with this sentence. Okay. And yeah, the thing that I was saying with the cyclic AMP production, okay, is also correct. It's also correct. ليش قلت خبصت؟ No, ما خبصت. It's also correct. The cyclic AMP production is also correct. So you can compare the cyclic AMP production with that of a sample having cholera toxin GTP and with one that doesn't, which is IV. Okay. What's it called? With one that does, which is IV. Okay, so you compare the cyclic AMP production of a sample without GTP and a sample with GTP. The sample without GTP mentioned that it's III. The sample with GTP mentioned that it's IV. Okay, mention it because it's in the marking scheme. Okay, part شو اسمه? Is this part C? Based on the data, describe the effect of cholera toxin. Okay. Based on the data, describe the effect of cholera toxin on the synthesis of cyclic AMP. Calculate the percentage change in the rate of cyclic AMP production due to the presence of cholera toxin in sample IV and sample II. Okay, so let's go here. What's it called? Based on the data, describe the effect of cholera toxin. Okay, so let's go one by one. Okay. So between samples, so if you look here between samples 2 and 4, so we're going to compare when GTP is the same because they want the effect of cholera toxin, they don't want the effect of GTP. So we're gonna take the sample I and III and we're gonna take the samples II and IV. Okay, because in I and III you have no GTP, in II and IV you do have GTP. Okay, which is why we're going to take those two and we're gonna compare them. ماشي? So for this we're gonna say that شو اسمه in sample I and III when we added the cholera toxin it had no effect. Okay, and in sample II and IV when we added the cholera toxin it increased the cyclic AMP production drastically. Okay, but that's only because you had GTP. Let's see. Cholera toxin increases the production of cyclic AMP in the presence of GTP, just like I said. Okay, and the cholera toxin has no effect on the production of cyclic AMP in the absence of GTP. Okay, basically all you have to do is if you're trying to find the effect of cholera toxin, GTP has to be the same. So you're going to compare the ones with no GTP and you're going to compare the ones with GTP. The ones with no GTP, when you added the cholera toxin, nothing happened. 0.5, 0.5, they stayed the same. The ones with GTP, when you added the cholera toxin, it went from 10 to 127. Okay? And yeah. So part D, a drug is designed to bind to the cholera toxin before it crosses the intestinal membrane. Scientists mix the drug with cholera toxin and then add this mixture and GTP to a sample of intestinal membranes. Predict the rate of cyclic AMP production in p mole per milligram adenylyl cyclase per minute. If the drug binds to all of the toxin. In a separate experiment, wait, part C. I didn't do the percentage change. Guys, we're grade 11, so we should know how to calculate percentage change. It's what's it called? It's the new minus the old over the old times 100. Okay, so you do 127 minus 10 over 10 equals to 11.7. Then you multiply it by 100 and you get the percentage. You get the percentage change. How do you know which one to put first, which one to put down, which one to put, what to put here, what to put there, what to put there? Basically here in part C they told you in sample 4 compared with sample 2. Okay, sample 4 compared with sample 2. If you're doing sample 4 compared with sample 2, that means sample 4 is the new one, sample 2 is the old one. Okay, like that's the best way I can put it. You're comparing sample 4 with 2, so 2 is like the original one. Okay, so like 127 minus 10 over 10 equals to 11.7 times 100. Why are you using these numbers aslan? Number one, they're the only numbers given to you and they want the rate of the percent change in cyclic AMP production, so that's why I use those numbers. Okay. A drug is designed to bind to the cholera toxin before it crosses the test. Okay, so where was I? Okay. In a separate experiment, scientists engineer a mutant adenyl cyclase that cannot be activated by GS alpha. Okay. The scientists claim that the chlorotoxin will not cause excessive water loss from whole intestinal cells that contain the mutant adenyl cyclase. Okay. So they said in the marking scheme an acceptable prediction that the rate of cyclic AMP production in p mole cyclase per minute if the drug binds to all the toxin is 10. Why? Go up. See here. GTP is there. Okay. GTP is there. What's it called? Wait. Predict the rate of cyclic AMP. Wait 1 second, guys. Guys, wallah, I don't know what's wrong with me. I'm so sorry. They literally told you that they added GTP. Okay, so you have GTP, and then you know that your what's it called? That your adenyl cyclase can't be activated. Okay, so basically you don't have cholera toxin. Okay, so you go GTP isn't there, cholera toxin, GTP is there because you added it. Cholera toxin isn't there. Rate of cyclic AMP is 10. Okay. Why is cholera toxin not there? Look, after the GTP binding, after ligand binding, GTP replaces the GDP, okay, to GS, and that is bound to GS alpha, a subunit of the G protein. Okay, this causes GS alpha to activate other cellular proteins, including adenyl cyclase, that converts ATP to cyclic AMP. The cyclic AMP activates protein kinases in the cells in the small intestine. A cyclic AMP activated protein kinase causes further signaling that ultimately results in this case of the normal GS alpha, GTP doesn't activating. Okay, individuals with cholera. This is due to the effects of the bacterial cholera toxin that enters intestinal cells. Okay, scientists studied the effects of cholera toxin on 4 samples of acid. Wait, then, how do you know that cholera toxin isn't there? Did they tell you in the question? Am I reading or not? Predict the rate of cyclic AMP. Okay, the drug binded to all the toxins, so it inhibited everything. Okay, that's why it's not there anymore. So cholera toxin is bye-bye. GTP is still there. You get 10. Okay. And just the justification, an acceptable justification that even in the presence of the toxin, cyclic AMP will not be produced. Okay. The protein kinases will not be activated, and Cl- ions will not be secreted. Okay, so if cyclic AMP is not produced, the protein kinases won't even be activated بالأصل. Okay, so if the protein kinases aren't activated, Cl- won't even leave, which is why we won't even lose water. Okay, so if the Cl- ions don't leave, we won't lose the water. Okay, based on the chemical structure of cyanide, identify one type of biological macromolecule that could serve as a precursor for the production of cyanide in plant. Justify your choice. Okay, so let's read. Secondary compound cyanide is a toxic bitter tasting chemical that is found in apple seeds. Cyanide in seeds is only released and tasted if the seed is crushed. When animals eat apples, they typically eat the sweet fleshy part of the fruit and spit out the seeds or swallow them whole. Basically, you need to know the structure of the macromolecules. Okay, so one type of biological macromolecule, we know that both proteins and nucleotides have both proteins and nucleic acids, they have nitrogen. Okay, so it can be either proteins or nucleic acids. You can choose either or. Okay, so part B, it has been proposed, and yeah, I justified it for you. So the macromolecule contains nitrogen. Okay, so part B, it has been proposed that apple trees are in a mutualistic relationship, okay, with the animals who eat the apples. Describe one benefit to each organism in the mutualistic relationship. Okay, so we need to identify one benefit. The benefit شو اسمه what? They didn't even write a benefit. Organism, plant, animal. Describe. Oh, okay. Yeah. So this when they say seed dispersal here, they told you that the animal eats the fleshy part and then spits out its seeds. So the animal disperses the seed of the plant. So the plant has the benefit. Okay. Then the animal feces provides nutrients for the plant. Obviously, that is what's it called? A plant benefit. And then the nutrient and energy acquisition obviously is an animal what's it called? Is an animal benefit because the animal is eating, so it's acquiring nutrients. Okay. 62. A major distinction between prokaryotes and eukaryotes is the presence of membrane-bound organelles in eukaryotes. Describe the structure and function of two eukaryotic membrane-bound organelles other than the nucleus. Okay, guys, we've talked about this like 20,000 times, so I'm not gonna go over this really. Pick, what's it called? Yeah, pick two of these, okay? You pick two of these, you explain the structure, then you explain the function. Okay, I have done this too many times. Okay, mitochondria, chloroplast, endoplasm. I've been doing it for the whole term. Okay, so please, guys, you should know this by now, yeah? Prokaryotic and eukaryotic cells have some non-membrane bound components in common. Describe the function of two of the following and discuss how each differs in prokaryotes and eukaryotes. This actually came in our final, in our last periodic, okay? I don't, it's really, well, actually, it's not highly unlikely because today we got the same written part as last mechanics final, so this can come as well. Okay, so yeah, this might come, not just because it came in the periodic last time, it won't come in the final, it actually might come. So make sure you know it. Here's the marking scheme for it. You guys should know this, you know. What should call it? And then explain the endosymbiotic theory of the origin of eukaryotic cells and discuss an example of evidence supporting this theory. This also came in both of our periodics. So if you guys don't know, then guys, wallah, I can't tell you, like, memorize it at this point, wallah. If you guys don't know by now, everyone should know. Okay, so yeah, that's it. So number 63. Identify four organelles that should be present in the eukaryotic organism and describe the function of each organelle. Great, I have to read now. During an investigation of a freshwater lake, an AP biology student discovers a previously unknown microscopic organism. Further study, okay, it's a unicellular organism and it's eukaryotic, so you can identify four organelles very easily. Okay, you know all the organelles for the eukaryotic organism: nucleus, ribosome, endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic, mitochondria, chloroplast, vacuole, whatever. Okay, choose four. Okay, you can choose four. Explain them. ماشي. Explain the function. Then prokaryotic cells lack membrane-bound organelles found in eukaryotes. However, prokaryotes must perform many of the same functions as eukaryotes. For three of the organelles identified in part A, explain how prokaryotic cells carry out the associated functions. So, for example, I said I want chloroplasts here. We know that prokaryotic cells, they don't have chloroplasts. Okay, so the thing that does it in prokaryotic cells, the thing that does the function of chloroplast, which is the light absorption, the photosynthesis, and the carbohydrate synthesis, is actually the thylakoid membranes. Okay, so here, look, you go down, you see the chloroplast, the light absorption, the photosynthesis, and the carbohydrate. Oh, okay, sorry. Part C. Where is part? What is it? Oh yeah, part C. Part B, sorry, part B. So look here, the chloroplasts, other membranes, thylakoid membranes, or other membranes, okay, function in light absorption, photosynthesis, carbohydrate synthesis. See, they don't even tell you to say thylakoid membranes. You can just say other membranes, you know. And yeah, that's pretty much it. Okay, if مثلا خلينا نقول, if you said the nucleus up there, okay, if you said nucleus, you can say the hereditary information, DNA, chromosomes, or RNA synthesis in the cytosol. So all the information of the DNA, your chromosomes, your RNA, they're all in the cytosol. Okay, so the functions of the nucleus are all carried out in the cytoplasm. And then ribosomes, if you pick ribosomes up, it's the same. It's just ribosomes. Okay, it's they both have ribosomes, so it's the site of protein synthesis. If you picked مثلا خلينا نقول endoplasmic reticulum up, you can say the diffusion of the molecules is in the cytoplasm because there's no endoplasmic reticulum, so it just diffuses على طول in the cytoplasm. Okay, see, the protein synthesis and the transport is in the cytoplasm. For the rough endoplasmic reticulum, for the smooth endoplasmic reticulum, same thing, it occurs in the cytoplasm. The lipid synthesis and the detoxification. Okay, So it's pretty straightforward, okay? According to the endosymbiotic theory, some organelles are believed to have evolved through a symbiotic relationship between eukaryotic and prokaryotic cells. Describe three observations that support the endosymbiotic theory. This is very similar to what came in our periodics. The past two is very similar to this. It's very similar to this, okay? So you can choose any of these three. The mitochondria has its own DNA. The chloroplast has its own DNA. The mitochondria can self-replicate. The chloroplast can self-replicate. The mitochondria chromosomes are circular. Whatever. Pick three of these. You know them. I've explained them too many times. Okay. 64. Describe the role of carbon in biological systems. So let's go here and what's it called? Actually, there's no need because okay, guys, we all know that carbon is like a major part of carbohydrates, lipids, proteins, nucleic acids, everything basically. Okay, so we can say that the role of carbon in the biological systems is to build the macromolecules, proteins, nucleic acids, lipids, and carbohydrates. Let's see the marking scheme. So the response indicates that carbon is used to build biological macromolecules. Wow, biological. You can just say macromolecules, such as carbohydrates, proteins, nucleic acids, and lipids. Okay? Now, the membranes of both B cells and the cancer cells are largely composed. Yeah, okay, now I think I have to read. Okay, explain how when the membranes are fused, the polar parts of the phospholipids from one cell will interact with the phospholipids from the other cell, and how the non-polar parts of the phospholipids from one cell will interact with the phospholipids from the other cell. Do I need to really read up? Let's see. The response indicates that the polar part of the phospholipids from one cell will align and interact with no. Okay, guys, this is just pure like basic knowledge of what's it called? Of polarity and of just polarity. Okay, so we know that the polar regions will interact with each other and the non-polar regions will interact with each other. That's all they're saying here. So the response indicates that the polar part of phospholipids from one cell will align and interact with the polar parts of the phospholipids from the other cell. Non-polar parts of the phospholipids from one cell will align and interact with the non-polar parts of the phospholipids from the other cell. Just like how we know that the non-polar parts of anything interact with the non-polar parts of anything else, the polar parts of anything will interact with the polar parts of anything else. Okay. And part C, make a claim about the most immediate effect on the fused B cancer cells if the fused cells are transferred to a growth medium that lacks a source. Okay, now, lacks a source of nitrogen. Okay, I hate reading questions. Lacks a source of nitrogen. Okay, what's it called? It won't be able to synthesize DNA or RNA because nitrogen is a part of nucleotides. You don't have nitrogen, you don't have nucleotides. You don't have nucleotides, which are the building blocks of DNA and RNA. What's it called? You can't grow. You can't what's it called? You can't grow. You can't synthesize DNA, RNA. You can't grow. Okay, and you can say the response indicates that the cell will die. Okay, the cell will die because you don't have you don't have nitrogen, so nitrogen is also a big part of proteins. You won't have proteins, you won't be able to grow. You don't have DNA, you won't be able to synthesize DNA or RNA. You're in a rough spot, you know. Okay, so the cell will die. Part D, provide reasoning with evidence based on the consumption of biological macromolecules to support your claim. Support my claim on what exactly? Guys, that's the thing that I said about nitrogen, because here they didn't tell you to justify. Okay, you just made a claim. Here they told you to make a claim. They didn't tell you to justify. Here they're just telling you to justify, because all you have to do is say the DNA, RNA, and the amino acids, the polypeptides, the proteins, whatever. Okay, they all contain nitrogen. So number 65. Number 65 is quite interesting. It has like brothers and sisters in number, here is it? Number 68 and but wait. No, there was more. No. Wait. 66. Oh, okay. So it has brothers and sisters. So it has 65, 66, and 68. Is the same question, but like different. It's like it's the same given, but different questions for some reason. I guess they love this question. Guys, listen, it's hard. Okay, it's like it's probably the hardest question, but I'm not gonna tell you guys it's not gonna come, because if they put three versions of the question, one is bound to come. So focus with me. I'll keep them to the end. Okay, so like we can go like all in one. Okay, so I'll keep them to the end, and I'll continue with the rest, and then I'll go back to 65, 66, and 68. Okay. Proteins, large complex molecules, are major building blocks. Blah blah blah. The chemical composition and levels of structure and of structure of proteins. Okay, so basically all you have to say is 4 points. Wow. Oh, okay. What's it called? You have to say a lot. Okay, so obviously the chemical composition is amino acids. Okay, so they're the basic building blocks of proteins. The other point, the amino acids contain amino carboxyl or correct structural formula showing amino carboxyl or proteins are composed of carbon, hydrogen, oxygen, nitrogen. What? Guys, I tell you to take 1 point from here and 3 points from here. Saraha, I feel like that's way easier. Just take 1 point from here and then 3 points from, because that makes more sense. I mean, like actually, no. Take 2 points from here and 2 points from here, which is that's actually way easier. You can just say that amino acids are the building blocks of proteins, and then say that proteins are composed of carbon, hydrogen, oxygen, and nitrogen, which I've been yapping about for all of the video. So carbon, hydrogen, oxygen, and nitrogen, okay, are proteins are composed of that, and amino acids are the building blocks of proteins. Okay. And then the roles of DNA and RNA in protein synthesis. Oh, no, wait. Where am I today? Okay, 3 points maximum. So you just choose the structures, I say primary and secondary. Okay, you just give the sequence. So it's just like a linear structure. Okay, and you give the bond responsible for it. They're linked by peptide bonds. And that's it. One of these is enough. I gave you 2. Don't do this. This is just confusing. Just pick one of these. This is annoying, for the love of God. Secondary structure, just say the alpha helix beta plated sheet. Okay, either or is fine. And then you can say that the hydrogen bond is the one that's responsible for the secondary structure as well. Whatever you want, but I say go with alpha helix or beta plated sheet. Okay, and then now we can go to the to part B, which is the roles of DNA and RNA in protein synthesis. So this one also has 4 points maximum. Okay, so information in DNA is transcribed to mRNA, which is transferred into protein. Okay, that is a role of DNA. And then DNA contains the information that ultimately determines the sequence of amino acids in the protein. Roles, DNA, it codes for RNA, it codes for mRNA, it codes for tRNA. Guys, I've done this so many times. Like, I'm genuinely just reading this for you. I don't know. Like, I've said this so many times. Okay? There's genuinely no need for me to go over this. You know this. You know all of this. Choose 4 points of it. Choose 4 of these. Okay? Choose 4 of them. You're fine. Okay? Choose 4 of them and you're good. Okay, guys? The roles of proteins in membranes in membrane structure and transport of molecules across the membrane. Okay, where is part C? Okay, so now the roles of proteins in membrane structure. We know that it's just 2 points maximum. You guys can go integral proteins, integral membrane proteins. Okay, and just say integral membrane proteins, and you can say receptors as well, or you can say the cell recognition, or you can say cell-to-cell communication as well because of the cell signaling. Just say receptors and integral membrane proteins. You should be fine. So the roles of proteins in membrane structure and transport of molecules across the membrane. 1 point for receptors and 1 point for membranes. 1 point for a description of integral or okay. I say then go with receptors and enzymes then. Yeah, just go with receptors and enzymes. Okay, that should be fine. Go with receptors or enzymes, and then what's it called? Take 2 points from here. So you took 2 points from here. You said the role. So you said the roles. So you said receptors and enzymes, and then you can take 2 points from here. You can say transport proteins may be specific. Okay, so there are transport proteins that may be specific, and the process may require direct input of energy. You know, when you put energy in, you put ATP. What's it called? You have to go from low concentration to high concentration, and you need a protein for that. So what's it called? So you could use the ATP. ماشي, and this elaboration, description of a specific transport system, la la la. You don't need this. Just take 2 from here, 2 from here. You're fine. Okay. Number 68. Oh yeah, number 68 is one of the brothers and sisters and family members, for the love of God. Wait guys, give me a second, something is happening. Okay guys, sorry, I had a little detour. Okay, so this I did it in my yeah I did it in the periodic video. So guys, you know what to do. Last question. So well, actually, it's question 70. I still have 65, 66, 68. Don't worry, I didn't forget. Ya Allah. Ya Allah. Why? Water potential in potato cells was determined in the following manner. The initial masses of 6 groups of potato cores were measured. The potato cores were placed in sucrose solutions of various molarities. The masses of the cores were measured. Okay. Again, after 24 hours. Percent changes in mass were calculated. Okay. Hear what I'm saying. Percent changes in mass were calculated. Graph the data. So the molarity, which is the independent, is on the X. The percent change in mass, which is the dependent, is the change of Y. This is what we measured. This is what we're changing. What we're changing is in here. What we're measuring is up there. Okay. You plot your points. You do your little line. We know it from the last periodic. We know how to draw. We know how to graph. So we're fine. Okay. What are the components of water potential, and why is water potential important for the movement of water in animals? So we know that the components of water potential are the pressure potential and the solute potential. Okay. The importance of water potential. So you only get 1 point for saying that it's pressure potential and solute potential. Okay, so be very careful. Okay, guys. So we know that water ensures water moves into plant roots. You remember by the by the root pressure. You remember how osmosis, by root pressure. Okay, so the water moves into the plant root by this thing called root pressure. You don't need to know what it is, it just does. Okay, so it ensures that so the water potential is actually related to the root pressure. Okay, so it ensures that water will move into the plant root. Okay. It helps of movements of water within the plant. Yes, we know that by cohesion and adhesion, it allows the water to move through the plant xylem smoothly. Okay. Factor involved in transpiration. Skip that. It's just stupid. Cell wall allows for increased pressure. This is good. What's it called? You know how when the water comes inside the cell wall will allow for an increase in pressure. This increases the pressure potential, which actually increases your water potential. Okay. And the pressure might counteract osmolarity. Yes, it counteracts osmolarity because osmolarity is always negative. Pressure potential is always positive. Osmolarity is the same as solute concentration, solute potential. Okay. And that's it. Okay, so you can pick 3 of these. You have to say pressure potential and solute potential though. Okay, and predict what would happen to typical animal cells placed in 0.0 molar and 1 molar sucrose solutions, and explain your prediction. So we know that animal cells, if you put it in a 0 molar sucrose solution, meaning that it's very high water potential, not very high solute potential, because there's no sucrose, there's no solute. Okay, we're putting it in a hypotonic medium. We're putting it in a hypotonic environment. That means that water will go from the high concentration to the low concentration, just like always. The high concentration is the environment, because it's a hypotonic environment, because there is no there is no salt. So it goes from the environment inside the cell. So the water comes inside the cell. Okay. If it's an animal cell, it might burst. Okay. And then they told us animal cell, okay, so the animal cell might best. And then the 1 molar sucrose solution, meaning there is salt outside in the environment. That means now the higher water potential is where it's actually inside our cell compared to the outside. The outside is actually less now compared to the inside. So the water will actually go from the inside of the cell to the outside of the cell, meaning our cell will shrivel up. Please tell me. Yes! Okay, guys. And yeah. That's it for this video. No, I'm joking. 65. A cell where... Oh, okay. Yeah, you guys don't know what's going on. Okay. Glycogen is a complex polymer of glucose molecules involved in the storage of energy in many organisms. Okay. Depending on the signals received by a cell, different enzymes are activated, which will lead to either the synthesis or breakdown of glycogen. The product of glycogen breakdown, glucose 1-phosphate, can be modified into glucose 6-phosphate, an intermediate of glycolysis. This is all haki fadi in my opinion. Okay. This is where the actual good stuff, like this is where you actually understand what's going on in the question. Okay. Make sure you understand what's going on in the question, because I'm not going to reread the question. Like I'm going to go 65, solve ABCD, 66 ABCD, 68 ABCD. Okay. So make sure you know what's going on. So in the liver, glycogen synthase and glycogen phosphorylase are regulated by kinases. They're regulated by these guys. So this is regulated by this and this. This is regulated by this. Okay? Okay. Hello. Now, these kinases transfer phosphate groups from ATP to the amino acids in glycogen synthase and glycogen phosphorylase. So these are transferring phosphate groups from the ATP molecules to this and this. So these guys are transferring phosphate molecules from ATP to these two guys. Okay? Then phosphorylase kinase B, PKB, is a kinase that regulates glycogen synthase kinase 3, GSK. This regulates this. This regulates that. Then they're going to tell you that this regulates this. Okay? Regulates the activity of and then this regulates this. Yeah, I said that. Phosphorylase kinase. Yeah, so this regulates this. That's what they're saying there. The activity of these kinases depend on the activation of signaling pathway. Obviously, the activation of these kinases, the PK, GSK3, and PKB, are dependent on whether this is actually activated or not. On whether they're dependent on whether the glucagon receptor or the insulin receptor actually get activated or not. Okay, and yeah, so let's go. By the way, guys, pay very close attention to this. So this هذا الشكل هذا, the T inhibits. Okay, so it stops. The arrow activates. So the insulin activates the PKB, which inhibits, which stops the GSK3, which stops glycogen synthase. This glucagon receptor will activate PK, which will activate glycogen phosphorylase. Okay, now this glycogen synthase will turn the glucose 1-phosphate to glycogen. Now, if we have glycogen phosphorylase, it will turn the glycogen to the glucose 1-phosphate, then the glucose 1-phosphate to glucose 6-phosphate, and then it will undergo glycolysis. Now you see, glycolysis, it's very interesting because it produces things that we've never heard of before in our lives, but I'll explain it to you guys, don't worry about it. So, a cell where PKB is always active has a mutation that results in the permanent activation of GSK3. So now GSK3 is always activated, okay? Based on the information in figure 2, predict the effect of this mutation on the activity of glycogen synthase. So we know that GSK regulates glycogen synthase, and we know that it actually stops glycogen synthase. So if it's inhibiting and this is always active, if GSK3 is always active, that means glycogen synthase is inactive because GSK stops the glycogen synthase. Okay? So I predicted it and I justified it. So this is the marking scheme. Glycogen synthase will be inactive. Role of GSK is to phosphorylate glycogen synthase, resulting in its inhibition. Inhibits in the presence of active PKB. And yeah, so make sure you write inhibition. GSK is to phosphorylate the glycogen synthase, okay? Basically to inhibit the glycogen synthase, okay? And yeah, so even if the PKB is always active, by the way, so this doesn't change what's it called? this doesn't change anything. We know that this is always active, so عادي. If this is always active, this is always going to be inactive. Okay. Describe the process that results in the activation. By the way, I hope you guys are paying attention to my mouse because I'm using it as like my pen, as like my underliner. Okay. Describe the process that results in the activation of multiple copies of PKB in response to the binding of a signal molecule of insulin to its receptor. Explain why insulin can stimulate the activation PKB but not the activation of PK. Well, second part is straightforward, and the first part isn't that hard either. Okay. So what's it called? We're saying they want, listen very carefully, multiple copies of PKB. Multiple copies of PKB. That means they're talking about what are they like hinting at? They're hinting at the signal transduction pathway. Okay. So, and where is PKB? It's not here. It's there. Okay. It's in the insulin receptor. It's with the insulin receptor. So the PKB is activated by the signal transduction pathway of the insulin receptor. Okay, is that what they say? In the signal transduction pathway activated by insulin binding, okay, each enzyme in the pathway can catalyze multiple phosphorylation reactions. Basically, in the signal transduction pathway that's activated by the insulin, okay, each enzyme in the pathway can catalyze multiple phosphorylation reactions. Okay, so it can undergo multiple reactions. Okay. Predict the effect of this mutation on the activity of glycogen synthase. So justify prediction. Predict the effect of this mutation on the activity of glycogen synthase. Insulin combined to the insulin receptor, but not the glycogen. Blah, 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 blah. Wait, no, part B. Okay, explain why insulin can stimulate the activation of PKB but not PK. Now here you have an insulin receptor, or here you have a glucagon receptor. How is the insulin going to activate PK if PK is activated by the glucagon receptor and not the insulin receptor? It won't work. But they have different shapes. See, an explanation that insulin can bind to the insulin receptor but not the glucagon receptor because the shapes are not compatible. Okay. Part C, explain why an increase in glycogen phosphorylase activity might result in an increase in O2 consumption in the cell. Guys, I'm going to make this so simple for you. Okay. Glycogen synthase. Wait, what did they say? Did they say glycogen phosphorylase? Yeah, glycogen phosphorylase. Okay, thank God. Glycogen phosphorylase. When you have this, first you start from here. Insulin, PKB, GSK, glycogen synthase. Okay. Here, you get glycogen. From the glycogen, your glycogen phosphorylase will convert your glycogen to glucose 1-phosphate. The glucose 1-phosphate will then be converted to glucose 6-phosphate. The glucose 6-phosphate will undergo glycolysis. Now listen to me very carefully. In glycolysis, you have a product called Ya Allah, where is it? What is the name of the product? Okay, it has a weird name. Where is it? No, there has to be. Where is the weird name? Okay, I'll tell you guys the weird name later. Basically, you have a product. Okay, this product. Okay, it's like what's it called? It's the product of this, the Krebs and citric acid cycle. It's involved in this. Okay, the Krebs and citric acid cycle. Basically, what you have here. Okay, you have this weird product. It goes into an electron transport. It goes into an electron transport channel, and in this electron transport channel, okay, O is the last receptor, is the last electron acceptor. So the whole electron transport channel is dependent on O. Is dependent on O accepting the last electron for it to be transported. If O isn't there, يعطيكم العافية. O has to be there. So if we have an increase in glycolysis, that means we have an increase in the production of this weird named product, meaning that this weird named product, more of it will go into the electron transport channel. The more it goes into the electron transport channel, the more O's we need to accept the electrons. صح. The more O's we need to accept the electrons, meaning what's it called? The O2 consumption in the cell will increase. I yapped a lot. Basically, all you need to say is the response explains that an increase in glycogen phosphorylase activity can lead to an increase in glycolysis, which will lead to an increase in the increase in the electron transport chain, which consumes O2. just like I explained to you in a very weird, long way, but yeah, that's what you need to know. A certain type of tumor results in the overproduction of glucagon. So now we have a lot of glucagon. Researchers claim that the treatment with insulin can counteract the effects of excess glucagon on the pathway, shown in figure 2. Provide reasoning to justify the researchers' claim. The response includes a justification that glucagon stimulates glycogen breakdown, but insulin stimulates the synthesis of glycogen. Let's see. Insulin. Here, look. Insulin, just like you see here, glycogen synthase, you're producing glycogen, okay? Only this glycogen phosphorylase, which is responsible for, which glucagon is responsible for, breaks down glycogen. It makes glycogen undergo glycolysis. Without the glycogen phosphorylase, we're fine. Glycogen stays. So the insulin makes glycogen. The glucagon is breaking it down. So if we said that the tumor, has a lot of glucagon, that the tumor causes an overproduction of glucagon, that means we have a lot of glucagon. That means we have a lot of glycogen being broken down, because we said that glucagon is the thing that's responsible for breaking down glycogen. So to counteract this, we're going to add we're going to we're going to add insulin. صح؟ we're going to add insulin. We're going to treat the cell with insulin. We're going to add insulin. Why? Because the insulin is going to produce glycogen. Meaning, glucagon destroys glycogen, and the insulin will make one. Glucagon destroys the insulin will make one. The glucagon will destroy the insulin will make one. وهيك وحدة بوحدة. Okay? And that's it. Guys, I feel like I'm so loud and so يعني مندفش على الموضوع. Okay. So same given. A cell has a mutation that results in the permanent activation of glucagon receptor even in the absence of glucagon. Okay. So the glucagon receptor is always activated, huh? Even if glucagon isn't there. Okay. Predict the effect of this mutation on the activity of glycogen phosphorylase. Glycogen phosphorylase activity, أكيد it will always be active. Okay. Now, to justify this prediction, the activation of glucagon receptor leads to the activation of glycogen phosphorylase, provided that there is not enough glucose 6-phosphate to inhibit the glycogen phosphorylase. Now listen to me very carefully. You see this added little piece of حاجة فاضي, okay? I'll explain to you like this, okay? So listen. We know, so they told us that this receptor is always activated even without glucagon. So this is always activated, ماشي? This is always activated, which is always activating this, the glycogen phosphorylase. Now, the glycogen phosphorylase breaks down causes the glycogen to be broken down into glucose 1-phosphate, which is then broken down into glucose 6-phosphate. But the crazy thing about this is that glucose 6-phosphate actually tries to stop the glycogen phosphorylase from breaking down the glycogen, because sometimes the cell can't keep up in a way. Okay, so like glycolysis is happening, it's happening. طيب till when يعني we won't have any more glycogen left. Okay, so we need something to inhibit the glycogen phosphorylase. We need something to stop this. We need somebody, which is this, to tell somebody, which is this guy, to tell this guy to whoa, calm down يعني. Okay, خلاص, we don't need any more glycogen to be broken down. خلاص, we're fine. Okay, so provided that there isn't enough glucose 6-phosphate to stop this, provided that since this is always active, there's so much of glycogen phosphorylase, okay, what's it called, provided that there's so much glycogen phosphorylase that the glucose 6-phosphate won't be able to inhibit all of it, it will always be active. Okay? Describe the role of ATP in the regulation of glycogen synthase activity. Explain how the addition of a phosphate group to... how long is my video? Oh my god, 1 hour, 10 minutes? Wow. Describe the role of ATP in the regulation of glycogen synthase activity. Explain how the addition of a phosphate group to certain amino acids of glycogen synthase can result in the inhibition of glycogen synthase. What am I reading? Describe the role of ATP in the regulation of glycogen synthase activity. Okay, now I understand it. Yeah. Okay, so guys, what's it called? where's glycogen synthase? Can result in inhibition of glycogen synthase. Okay. So wait. Okay, I got it. Okay. So PKB and GSK. So look, okay, listen. This is what we're talking about: glycogen synthase. Okay. Glycogen synthase is regulated by what? GSK. GSK is regulated by what? PKB. PKB is regulated by what? This, the activation of this. Okay. طيب. This is activated. When this is activated, what happens? It activates. It activates this PKB. صح? The PKB is carrying what? They told us that it carries ATP. صح? It carries the ATP from here to here. Now this is carrying the ATP from here to here. Okay. We have the ATP here. ماشي? We have the ATP here. Okay. The ATP got here. The ATP splits itself now. It goes into ADP and P. ADP and P. Okay? The P binds itself to the glycogen synthase. See, not glycogen phosphorylase, glycogen synthase. This is going to change the active site. This isn't going to make the active site better. It's going to make the active site worse because there's no phosphor. Why? Okay? Why are you adding this to me? You're damaging the active site. So then everything is going to be messed up. Okay, that's like the simplest way I can put it to you. Let's see what they put exactly. So yeah, that ATP provides the energy. Yes, so the ATP is obviously the thing that's providing energy to make the phosphate to bind the phosphate group to the glycogen synthase, just like I told you guys. Okay, so the bond is being formed. Just know that ATP is the thing that's providing the energy for it, just like I told you. Then the justification is so the addition of the phosphate group to the synthase could alter the structure and or change the glycogen synthase. It can alter the structure such that the substrate can no longer interact with the active site because the active site changed. The enzyme can no longer catalyze the reaction. Okay, so that's the best way I could have put it. Okay, so part C, explain why an increase in glycogen phosphorylate activity might result in an increase in... Okay, yes, okay. This is the weird product that I was talking about, okay? Pyruvate. What a beautiful name, bro. Stupid. Okay, so if we have a lot of this glycogen phosphorylase, we are breaking down a lot of glycogen to glucose 1-phosphate. This is going to be converted into glucose 6-phosphate. We're going to have a lot of glycolysis happening. We have a lot of glycolysis happening. A lot of pyruvate will be, will be produced, okay? Yeah, because an increase in glycogen phosphorase can lead to an increase in glycolysis. Okay, so because the glycogen phosphorase activity, increasing glycogen phosphorase activity will increase glycolysis. Okay, part D, diabetes often results in the production of reduced levels of insulin. Researchers claim that a drug that binds to and blocks to the active site of GSK3 might mimic the role of insulin. Okay, in the pathway shown in figure 2, provide reasoning to justify the researcher's claim. Okay, so let's go see here. Both insulin and this drug will prevent the binding of substrate to GSK3, effectively inhibiting the enzyme. The drug mimics the activity of PKB, which is activated by insulin. Okay, so look what they told you here. The diabetes often results in the production of reduced levels of insulin. Researchers claim that a drug that binds to and blocks the activity site of GSK. طيب. What binds to and blocks the activity of GSK? PKB. صح؟ طيب. What regulates PKB? Insulin. That's what regulates PKB. It's insulin. So obviously the GSK will mimic the role of insulin. The what's it called? That the drug will mimic the role of insulin because the drug inhibits GSK. If it inhibits GSK, that means it activates the PKB. And if PKB, PKB will hala. If PKB will be activated, that means the GSK will be activated. The GSK will be inhibited. If the GSK is inhibited, it will mimic the role of insulin. Okay, to justify the researchers' claim. And yeah, so both insulin and this drug will prevent the binding of substrate to GSK, inhibiting the enzyme. Okay. Both insulin and this drug will inhibit the enzyme GSK3. You can say that, I think. Okay, yeah, you can definitely say that. And the drug mimics the activity of PKB, which is activated by insulin. Yes, the drug does mimic the activity of PKB, which is activated by insulin. Why? Insulin is the thing that activates they said that insulin insulin inhibits GSK3, and what also inhibits GSK3? PKB. Okay. And number 68 now, finally. La Allah, A, B, C, D. A cell has mutation that disrupts the insulin binding of the site of the insulin receptor. Based on the information in figure 2, predict the effect of this mutation on the activity of GSK3. Obviously now the activity of GSK3 will skyrocket because there's nothing to inhibit GSK3 anymore. There's no insulin because the active site is changed. معناته insulin can't bind to it anymore. PKB won't be regulated. GSK activity will skyrocket because PKB was the thing that was keeping GSK3 in the down low. Okay. Yeah, that's basically what I said. Okay, so that's the marking scheme. Basically exactly what I said. B. Describe the role of ATP in the regulation of glycogen phosphorylase activity. Explain how the addition of phosphate group to a certain amino acids of glycogen phosphorylase can result in the activation of glycogen phosphorylase. Okay, now describe the role of ATP. It's the same thing. ATP provides the energy to form covalent bond with a phosphate group and glycogen phosphorylase. However, this one actually is better for it because it's glycogen phosphorylase. You're adding a phosphate group. That means you're making the active site more compatible. The active site is now more compatible. Okay? Meaning that it will be able to catalyze the reaction better. Okay? So phosphate groups alter the structures and or change of glycogen phosphorylase such that the active site is more compatible with the substrate. The enzyme can catalyze the reaction. Okay, because we're adding the phosphate group to glycogen phosphorylase, we're making it better. But when we add the phosphate group to glycogen synthase, what happens? It becomes worse. Okay. Explain why an increase in glycogen phosphorylase activity might result in an increase in ATP production in the cell. So explain why an increase in glycogen phosphorylase. Okay. The response includes an explanation that an increase in glycogen phosphorylase activity can lead to an increase in glycolysis, which produces ATP. Guys, I will give it to you very straightforward. Okay, ATP is one of the products of glycolysis. Okay, and if we have an increase in glycogen phosphorylase, we have an increase in glycolysis, we have an increase in ATP production. Okay, simple as. Researchers claim that even in the presence of high levels of glucagon and the absence of glycolysis, glucose 6-phosphate cannot be overproduced. Provide reasoning to justify the claim. Why can glucose 6 not why is glucose 6-phosphate never overproduced? مع انه they said that glycolysis isn't happening. Okay, absence of glycolysis and high levels of glucagon. يعني you're having a lot of glucose 6-phosphate being produced because you're having a lot of glycogen being broken down because of the glycogen phosphorylase. Okay, because look, when you reach the total amount of glucose 6-phosphate that the cell can hold, the glucose 6-phosphate will just start to inhibit this. It will just start inhibiting the glycogen phosphorylase. And since there's so much of it, it will always keep inhibiting. It will keep inhibiting, which means it can't really be overproduced. Okay? It's always being used up. And that's the marking scheme. Basically exactly what I said. Okay? Those are the written part questions, guys. I know they're long, but I tried my best to explain everything so you guys can actually genuinely understand. Make sure that that you study really well for tomorrow. Course practice, course revision, the quizzes. If you haven't done all of that yet, okay, I recommend sticking to the course revision and to the AP classroom quizzes, okay, and make sure you look at my chapter 4 summary video. Make sure you know that, because chapter 4 is very important for tomorrow. I have a feeling that most of the exam tomorrow will be chapter 4. Those of us who did the biology CA in the last week when nobody was there, we're very lucky because it was about chapter 4, so tomorrow it's going to be very easy. Inshallah ya Rab, tomorrow is easy. We'll be familiar with it. So I hope I hope you guys still remember your answers, because it might come copy paste. You never know. Okay. Thank you guys. Have a nice day. Study well. Okay. And good night.